and we're responsible for collecting, processing, testing and distributing all fresh products across the country, as well as plasma for fractionation, which we then provide to CSL Behring in Australia. Those products then come back to us and we distribute them out to Australian health providers. Sorry, Anne, but not moving on. There we go. Yeah. Sorry, I had the cursor over there. So look, this is just a summary of who we are and what we did in the financial year ending June 30, 2024. You can see we've got over 100 donor centres across the country. Where we collected over 1.5 million donations from over 500,000 donors. All our processing and testing is carried out centrally at one of those processing centres, either in Brisbane, Sydney, Melbourne or Perth. We also have, if you're looking carefully, 2 human breast milk processing centres. We started that as an initiative a few years ago. One's based in Sydney, one's based in Brisbane. And we now, as of just a few weeks ago, with the agreement with the Victorian government, essentially provide pasteurised human breast milk for premature babies across the country. A more recent development is that we've moved into fecal microbiota production. We have a GMP toilet in Perth. For those who don't know what fecal microbiota is, it's essentially poo transplants used. It's really almost now the first line of defence for multi-drug resistant. What's the name? But
CDID.
Immune system.
CDID.
Bacteria.
Oh, CDID, yeah, Clostridioides difficile. Thank you. So we're sort of moving through that. You'll also see on the right-hand side of that table that we provided just under 850 tonnes, that's 850,000 litres of plasma to CSL Behring through that last year. Our target this year is about 900 tonnes. We've talked a bit about plasma and the demand and the risk essentially to supply plasma across the world. That 900 tonnes, I was staggered when we sort of saw those numbers initially. We used to be self-sufficient in Australia about 15 years ago. We're only now about 48% self-sufficient. We have an increasing demand of about 7% per year, whereas our increasing collections is only about 5% per year. So it's a key focus that we're working through to try to address that. So you can see prior to COVID, we saw a steady decline in blood usage, as seen throughout the world. Introduction of patient blood management, change in medical practice and so on. But since COVID, we've seen an increase and we're really back to demand levels, pretty much what they were back in 2014. I know we're talking about code orange or yellow, I think, here in the UK. We've certainly had our ups and downs, but I think really we've been focusing very much on whole blood collection and we sort of run a daily inventory management system in the country. And I'm pleased and proud to say I think we've actually had quite a good response and no real problems with whole blood collections and supply for the last 12 months or so. As I said, plasma is an increasing issue for us. What we're doing is we're still trying to determine the drivers for the increase in demand. We ask our end users, we ask various different groups, anyone who's got any ideas about why there's this increase. It's not purely related to return to surgery that was sort of it was deferred during COVID. But we think there's a number of different issues. Some of it's the ageing population. People are now living 10 or more years longer with new cellular therapies for cancer, for example. There's an increasing demand for products, particularly IVIG again. I think that's one of the sort of real issues, I think, in terms of the future is with CAR T cell therapy, for example, the need to sort of supplement people with IVIG subsequently. But I think increasingly in Australia, we've also had a surge in immigration post-COVID. post opening our doors. I think it's become a political issue as well in our country as well as elsewhere. I think we had 500,000 net migration the year before last. A lot of them are students, a lot of are working holiday makers as well. But particularly a lot of immigrants coming into the country from our near neighbours. And so that's sort of changing the demographics of our country so that now, and I'll comment on this again later, about 30% of Australians are born overseas, and about 50% or more now have parents or who have been born overseas, compared with, well, when I was born, the majority of people that looked pretty much like me. There's been a significant change, and then that change is also reflected in a significant change in demand for the different blood products in the country. So as I said, we need more plasma than ever. Move on. So there's more and more uses for plasma being found. Essentially, it takes up to 15 plasma donations to make a number of different treatments for patients. And in Australia, we need to get at least 18,000 donations every week. I don't know if you recognise this guy. This is Michael Klim. Most people of my age anyway, and probably down to, I think the younger generations have got no idea who this is, but this is Michael Klim. He was a world record and an Olympic swimming champion back in the late 90s and 1990s and 2000s. A real star of Sydney Olympics in 2000. He, though, unfortunately for him, in 2020 was diagnosed with CIDP, it's chronic inflammatory demyelinating polyneuropathy, for those who want to know the full extent of it. But essentially, Michael depends on IVIG infusions every 6 weeks, essentially to keep him alive and to be able to sort of go about his normal day-to-day things. And interestingly, Michael lives in Bali now and actually flies into Australia every 6 weeks or so to get his IVIG infusions. What I also thought I'd put in here too is it's topical. I think we've made some comment briefly this morning as well. This is the man with the so-called golden arm, James Harrison, who died earlier this year. He's been making sort of worldwide headlines. New York Times ran a great story about him. Coincidentally, I saw in ISBT News just today a story about him that we put together out of Lifeblood. James has been, he's donated 1,173 plasma donations through his life. And his donations were used to produce anti-D, which is used, as most of you are aware, is used to prevent hemolytic disease of the fetus and newborn. And up until recently, and all Rh-negative women in Australia, but we've just also got approval with prenatal testing to determine the RhD type of the fetus, so that also reduced the demand. But this is just another example of the huge demand on our donors, and particularly in this instance, anti-D donors. We've got a pool of around about 100. We increased that to about 130 over the last couple of years. These donors volunteer to be infused with RhD positive red blood cells and generate an anti-D response, and then we bleed them and manufacture anti-D for distribution. So that's still the common treatment for HDFN, or prevention of HDFN, but which leads me on to some of the research that we've been doing. And 30 or more years ago, there was a lot of work in terms of trying to develop a monoclonal antibody for the D antigen, and there are a couple around, but not entirely successful. I think that's a fair comment, isn't it, really? But we've been working with a group at Walter Eliza Hall Institute in Melbourne and our anti-D donors to use novel technology to essentially isolate the individual cells that produce the antibodies in our anti-D donors, get the genes for those cells that produce those antibodies, clone those genes, manufacture them up, and then make those antibodies that those donors make themselves in the laboratory. So far, as you can see on this slide, we've actually been able to identify 16 separate monoclonal antibodies that we've put called up that actually are effective in the lab. So this is research, still early stage research, but we're looking potentially to reinvestigate the opportunity to develop a synthetic anti-D to move on. So watch this space, which essentially that's sort of which is my segue into our R&D program at Lifeblood. This is a slide that I put up for our management, essentially to sort of convince them of the importance of research. Essentially, we do add value. It's not a cost. It's an investment in the future. I don't need to tell people in this room here, I don't think that. And we have a broad program of research that covers essentially everything from the motivational aspects of donation, which we were talking about this morning, right the way through to new products that we can develop from our donors' products. So we've been underpinned by this research program, which I've been proud to lead for about 15 years or so. Our research is really focused on creating new knowledge, but importantly, translating that knowledge into practice as well. We talked a bit about that this morning as well. And I think that's my background. I've sort of worked in academia. I worked in CSIRO, the Commonwealth Research Labs in Australia. I spent 15 years of my life working in the private sector as well. So I've sort of got those various different aspects. The great thing about doing research in a blood operator is that you can actually translate the stuff that you're doing, use that technical term, into practice almost straight away, because you're sort of working with handing, as we heard this morning, essentially handing glove with the end users. So that's really what I think excites me about what we do at Lifeblood. So I won't go. I'll describe the various different areas there. I won't go into that anymore here. But I just wanted to sort of re-emphasise that our research program is strategically aligned essentially to help fulfil our purpose of collecting life-giving donations for life-changing outcomes. This diagram is our strategy 2027 in a nutshell, essentially our 4 key strategic areas, but essentially various different areas in there I'll refer to as I go through my talk today. I do apologise, man, about this one, but actually Alison made this one for me, but it serves a purpose really. It's really just to show you that we've got something like 60 to 80 different research projects. Each one of those little bubbles is a research project. The different colour codes to sort of match the different areas of research that we're working in. And you can see that going across from the left to the right on this screen, sort of early stage research, exploratory research, right the way through to translation into change practice or policy. I should say that while I can hear the bit at the left, we have a growing number of students as well. So any students in the room, there's opportunities really that we find to actually get into those more exploratory areas of research that some of us more, Lifeblood employees, we have to be much more strategically focused on doing things that we need to get sort of out the door tomorrow. You can see up on the left-hand side there the strategic areas of interest at Lifeblood where those research projects fit. I just thought I'd also include, sort of made a few last minute additions to this presentation last night, just to sort of give you an idea of some of the new projects that we've got underway before I get into some of the donor research. But and the reason for doing that is I think one thing that's really exciting, I think it was sort of in the similar context to what Richard was Stephen's talk this morning in terms of doing a very large study and having essentially the outcome that you may be not quite excited about, but it's still a good outcome. And that was our so-called CLIP2 trial. This is looking at the use of a frozen platelet instead of a room temperature stored platelet in cardiovascular patients. And I'll tell you a little bit about that in a minute too. But these are just some of the clinical trials that we've been working on. So essentially looking at new extended uses of platelets. I think I probably don't need, I'll say it anyway, but a platelet in Australia has a shelf life of 7 days. So we're a very large country. Moving platelets from Sydney, Melbourne, Brisbane or Perth out into the remote areas is a tricky issue. There was one instance I can remember about a couple of years ago Darwin, for example, right up in the top of north of Australia, had a patient in need of platelets. I think it cost us a charter flight essentially to ship a couple of platelets up to him up to that hospital from Brisbane. So wouldn't it be good if we could sort of have an alternative way of storing those platelets? One of these is essentially it's going back to the future, if you like, storing them at 4 degrees. We've got some great data from our labs in Sydney that show that if you keep a platelet rocking gently at room temperature for 4 days and then put it in the fridge, you can keep it in there for another 20 days or so, and it's still biologically active in the laboratory at least. So that's fine, and we published that work. So what we also wanted to do though was to see whether it's still effective in a clinical situation. And so this is what this study is about. It's the so-called CHIPS study, looking at cold stored platelets in cardiovascular patients. Looking at it's a randomised, non-blinded, non-inferiority study comparing normal issued platelets, that's room temperature 7-day storage, versus cold stored. That's sort of put in. We've got data to show that they're effective in the lab till 20 days after storing, and that trial will be reported on at the GAICD meeting, I'm told, in Milan, which is happening in about 6 weeks' time. So can't tell you the outcome, but we're hopeful it's completed and analysed and will be reported shortly. This is the one that I wanted to mention, Stephen, is the CLIP trial. CLIP 2 is a randomised controlled trial looking at frozen stored platelets versus room temperature platelets. And this arose from some work that we've been doing in our labs in Sydney, 15 years or so, 14, 15 years, on request from the Australian Defence Force initially. Sort of was around about the time when the Afghanistan war was still raging. We had troops on the ground over there, and we were relying on US and Dutch, primarily Dutch blood bank, essentially, to provide frozen materials for our troops on the ground. The Dutch pulled out. We thought, well, or our Defence Force thought, well, we're quite vulnerable here. Let's look at alternatives. And cut a long story short, we did some work with the Australian Defence Force to develop for them a frozen red cell plasma and platelet inventory. That's been done. And we've subsequently then run clinical studies in a civilian setting to compare frozen stored platelets. These are frozen at minus 80 degrees and then reconstituted at the bedside in the hospital to patients undergoing cardiothoracic surgery. I'll quickly run through these data. This data was completed. Why did that go away? I won't go through all the detail, but essentially, it's, as I said, similarly, it's a randomised, blinded, non-inferiority study and looking at number of bleeding 24 hours after surgery. These black things are coming up. Doing. Essentially, what we showed was that the frozen stored platelet was non-inferior to the fresh platelet, which was good. But what we also showed was that if you look at the secondary, and the non-inferiority was no worse than 30%, as good as the other one in terms of bleeding after 24 hours. What we did show, though, was that, so there's the non-inferiority data there. What we did show, though, was that with secondary outcomes, and that was improper blood loss, further measures of blood loss and usage and blood component usage in those particular patients. Essentially, I'll put it up on the next slide. The conclusions were that while the study met its endpoint of being non-inferior, essentially the product is probably 30% less effective than the current product. So once again, Stephen, we think it was an exceptionally well designed, very, very well orchestrated trial. We've got a lot of data. We presented this at the in October last year. It's still in the final versions. I don't know why we haven't actually sort of agreed to publish it yet, but it's still going through not editors, so actually authors changes. So, but we will have that published soon. And I guess that's the issue here as to what do we do now? And that's a question that we ask the Defence Force. Yes, it's a product that they want to have, but we haven't really got an answer as to whether we would have that as a product or issue.
Is the computer behaving, or did something disappear for you?
It was me, probably. I was pressing one of the wrong buttons.
Yeah.
Is it not going through?
As long as it's going out. It's
Yeah. No, it's your notes. Were they coming up?
Yeah.
Oh, they're coming up.
Yeah.
Perfect. Thank you.
If now I tried using the forward button on the keyboard. I didn't like that. Right. Moving right along. So I want to move on to our donor research program. And this really is just a snapshot of the program, what our team's working on at the moment. So I don't need to tell this audience, really engaging donors really is multifaceted. So we've got research projects really spanning quite a range of topics. The priority areas are shown here, and the priority areas I've listed are ones that have been put together by our organisation, strategic objectives for the whole organisation. So our donor experience team, our marketing team, and so on. We work very closely with those groups so that hopefully, to contrast some of the comments I made this morning about AMT, hopefully we will be able to translate all of the outcomes of our research programs into those environments as quickly as possible. So I want to go into a little bit more depth now with one of our projects. I might skip through quite a bit of this, but I just want to go through with you just ideas. So this is a Donors Recruit Donors project where we're really assessing the opportunities to use word of mouth techniques to recruit and retain donors across some of our changing demographics of our population. So I've noted up here the key. There's a picture of Barbara at the top, for those who don't know her. Barbara Kathleen Schell on the left-hand side is really the lead investigator in this project, and Michael Polonsky from Deakin University, who's Professor of Business and Marketing. So we essentially work together on this team. This is I should have said earlier, our research program at Lifeblood is actually funded as part of our program of support from Australian taxpayers and the government. This one, though, we were able to leverage some of the funding to win another research grant from the Australian Research Council. So that's essentially a bonus for us to get that extra funding. Project really, once again, aligns with one of our key strategic objectives, where we're aiming to have donation essentially being a respected and normal part of the Australian community. So we're sort of looking at how do you get people engaged? How do you do this? So that's one thing. Our marketing guys are working really hard to do that. This is really sort of trying to work through that as well. And we're really trying to increase the ethnic diversity of our donor panel so that it is truly representative of the changing face of Australia. So why do it? Because you might think, I thought as well initially that you talk to somebody about something, word of mouth, it's a pretty good way of communicating. We all do it one way or another. Why not use it as a way to recruit blood donors? So probably not surprisingly, there's very little literature in this space, and that's really it's essentially a social intervention. But there's wanting to understand how I actually can use that social intervention to actually activate donors to do something, and really it's aiming then to use active donors to use their networks to sort of broaden their networks to bring others into the fold. It has been reported as some of the most cost-effective donor recruitment strategies available. We don't need to provide incentives. You're just telling somebody how good it is, not on the barbecue or the water cooler or on social media and saying, well, this is what we do. Isn't this a good thing? So it is really considered a reliable source of information because you're dealing with people that you know generally, and it is important within an altruistic service context, given essentially this intangible, complex, and high risk nature of blood donation. Essentially, it's something that hopefully it could help us make blood donation be viewed as normative behaviour for those groups that are involved. Now, as I said, that while this might seem to be a no-brainer, there's surprisingly little research in this space when we commenced this with this project. And we really lacked the information to resolve this disconnect between intentions or willingness to donate and actual action of actually getting into the donation centre to do it. And importantly, we lack that sort of information in different ethnic parts of the community as well. So that's that's the raison d'etre, if you like, of doing this. So to address those gaps, we use this. I had looked this one up. I must admit, it's not being psychology thing, but it's a motivation, opportunity, ability, behaviour model. Really to sort of put some science behind the whole work here to do this. And we figured that would be able to highlight the multiple factors that interact to shape the individual's actions. The aim then was to examine that interplay of motivation, opportunity, ability, and behaviour, so that we'd be able to develop more targeted and effective interventions that we can then put in place for the operations side of the business. So the project really fulfills 4 distinct yet interrelated objectives that I'll list here, which I won't go through. But these all address the overarching aim of understanding what factors contribute to donors talking about blood donation and translating those findings into an increased participation of different ethnic groups in the Australian Blood Donor Panel. Now, I've talked about ethnic groups. What I mean by that is it's a group of people, obviously, within the community who have a different racial or ancestral background to the main population, with the majority of Australians still being white European. And we also sought input from our red cell reference groups responsible for actually matching donor and patient red cells with transfusion fill. They told us that the key areas we wanted to be looking at were retention and recruitment and retention of donors from the Asia Pacific region, so we have a large influx of people through there, due to a short supply and increased need of blood products from those people with these ethnic ancestry. So this is a summary of the overall project plan, and please, I'm not going to go through all the detail, but I'm more than happy to discuss this afterwards or put you in front of us. Some of this is actually in the final throes of preparation for publication as well. So we started with, and I think primarily, just to give you if you just quickly glance across the middle line, across from left to right, just the way we went about this was really just to start off with focus groups of our lifeblood staff to sort of get their perceptions, working with donors at the front line and where they're going to have to be the then have focus group with blood donors from the different groups we're working on. Use that information to feed into broader surveys to get sort of more information, bring that back and then co-design various different opportunities, interventions that we could test in laboratory or field positions, trial that with the various different blood donors, and then move that on to blood donors. And that was the ambitious timeline that we set up back in 2021. I'm pleased to say we're pretty well on track, and I'll talk to you about some of the data that we've got coming through now. So first of all, our blood donors focus groups. And we found, I'm not going to, for the study, we conducted 9 focus groups with 56 donors, recruited from 3 ethnic groups. So there were British Irish, that Irish control group in this one here. We had 20 Chinese and 20 Indian. They were the different areas that we're told that we should be looking at. And also quite relatively easier to access within our donor pools. The focus groups were facilitated by researchers who themselves were donors from those same cultural backgrounds to focus to the focus group participants so that we could enhance cultural safety and participant engagement. No, we're really engaged. Actually, just two different people that ran those two groups for us or with us. I'll quickly run through some of the insights from those focus groups. So, although our donors were willing and generally confident to talk about their own donation experiences if asked, they identified several risks associated with talking about blood donation. Most common was the fear of being seen negatively for boasting or bragging about the donating blood, or being seen as being too pushy in advocating for blood donation when blood donation really is considered a personal choice. Furthermore, some of the donors were concerned about how people would respond once the topic was brought up. Word of mouth by nature invokes a response. So donors were cautious, though, not to put others in a position where they would need to disclose a health issue or some fear of previous bad experience that they may have had with blood donors. Or indeed, whether they might be asked a question outside the realm of their own experience. For example, a woman asking a man whether she could donate while menstruating, for example. One reason people don't talk about blood donation is the lack of prompts or the limited triggers to start conversations. So about half the participants in these focus groups shared a post online about their donation, but fewer than 20% of them initiated discussions about blood donation. So most conversations are generally triggered by schedule questions such as, what are you doing this weekend? Or maybe a prompt, for example, what's that bandage that you've got on your arm? Donors are looking for a license, essentially, to talk proactively about blood donation without being seen as too braggy or pushy. They also want more triggers and prompts to encourage others to ask them. So once the conversations were started, topics included the donation process, their experience, recipients, eligibility and benefits to the donor. Donation experience, knowledge and access to resources influence the donor's confidence to talk about blood donation and encourage others to donate. Now, in contrast to blood donor staff, and I didn't go through the detail, but this is study 1A, non-staff blood donors did not want more information. Rather, they wanted the same information that they currently get, but in a different and simplified format. We tend to medicalise lots of this stuff, and it's second nature probably to all of us in this room, but it's not to the broader population. So donation and experience and knowledge of blood donation experience contributes to donors' ability and willingness to talk about donating so that they don't feel silly or sort of talking about something that is not essentially in their normal vocabulary. So essentially what we've learned here, providing donors with access to a variety of easily accessible and culturally relevant resources is really the key thing that came out of this group. Most of our donors recognise that engaging in word of mouth, the more people donate, the more lives are saved. But however, the donors' word of mouth really varied in agency, who initiates the conversation, goal, what they're talking about. Most donors were more reactive, that's responding to comments or questions by others, than being proactive. So it's a case of providing those triggers or stimulation to get the conversation going. Further, some donors were only willing to share information about blood donation of their experience, while others were willing to actively find answers and encourage donation. So these distinctions highlighted the 4 word-of-mouth roles that our donors engage in. So there's advocate, supporter, sharer, and responder. So the different categories which we've identified through those areas. So the specific role of those different areas really depends on the donor's donation experience, the context in which word of mouth was occurring. For example, online or offline, an opportunity for longer conversations, and their relationship with the people that they were talking to. Now, when encouraging blood donors with Indian, Chinese, or British Irish ancestry to talk about blood donation, a few substantial cultural considerations really needed to be considered. blood donation is viewed very differently between these groups. And this affects how and when it's talked about. For example, donors with Indian or British Irish ancestry, donors often refer to blood donation as a personal health topic, and their fear of health issues being shared as a reason for not donating blood makes it difficult for them to talk about it. For donors with Chinese ancestry, blood donation ranks low in their preferred pro-social cultural behaviour because it's linked to low health and vitality. This link to health makes talking about blood donation with people perceived or known to hold strong cultural views, for example, older people, it's difficult and prevents some donors from talking about blood donation with close family members and older members of their families and their community. So now, probably obvious too, but donors suggested that access to translated material and addressing those cultural misconceptions and culturally appropriate resources would improve their conversations. So finally, donors of Indian and Chinese ancestry noted that promoting the opportunity to donate in groups and highlighting the ethnic diversity of staff at collection sites would facilitate their engagement in word of mouth because they could then be more confident than the donation experience. I'm going to skip over a whole bunch of other slides now because I think there's some key messages from that first part. But I think the other thing I was just going to mention, so we talked about just those three different ethnic groups. We've also been doing some work on the Indigenous community in Australia as well. So I talked about socially and culturally responsible and acceptable things. Now, this is a whole new kettle of fish, really dealing with thousands-year-old concepts and also hundreds of years old aversions, if you like, to the medical community, to colonisers, if you like, in Australia. So it is a real issue for us, those areas. I'm not going to go through this well, but essentially the outcome of those focus groups, I hope that didn't bore you too much, but I think that I just want to give you some of the detail there, and I'll skip through the next bit. But from those, we've made recommendations then already to our donor experience and marketing teams. We use those basics then to go on to perform our surveys. We put focus groups in place, and I'll just skip quickly to what we do have, where we're up to right now, and that's essentially we developed a number of, you heard me talk about wanting more information about, so education about what's involved, putting it in terms that are not too medical to be able to be understood. So we've developed a number of e-learning modules with a number of our volunteers at Lifeblood. We practice with our volunteers, and you'll see the picture on the very left, the small one. Our CEO, Stephen Cornelison, I convinced him to actually be the spokesperson for this whole program. So he sort of introduced the whole concept. So once again, going back to translation, being strategically aligned, getting the CEO on board, I think was important. Certainly from our perspective, but I also think when we get to the stage of putting this into place in our donor centres and elsewhere, if the bosses are saying it should do it, then it helps. I think that's my message. So what's next is essentially just summarised down through here. So I will go on. I did want to talk about a couple of other things. This one's our Australian blood donor study. And this is sort of small biggies from what's already been happening over here in Manipulation as well. But I just wanted to give you some idea of what we're doing in Australia as well. And it also fits into Lifeblood's strategic intent to play a greater role in healthcare in Australia. At the outset, we're essentially contracted by governments to collect, process and distribute blood. If I were giving this talk 10 years ago, that's basically where it would end. Essentially, we're almost treated as a contractor and you don't get involved at the other end, just in your middle bit and go on. We've had a change in government, change in organisation, and we've had the pandemic. And I think it's finally dawned on our governments and others that a national blood operator, such as ours and yours, can and does, you do, because you're part of the health system, we're not. Can and does play a significant role in population health. I think there's some comments about donor apps and having donor information this morning. We are developing those, and we do have information on the donors' apps about, obviously, they've got their blood pressure, their hematocrit, all those sorts of things from time to time. We're looking at adding other things into those as we go along. And we did play quite a big role in population health through the pandemic, which I won't go into today, you'll be pleased to know. But I do want to talk a bit about this study. As I said, there's a number of studies such as this that are summarised in this table. There's a trade study right up there on the top line. But we went about, we didn't know whether something like this would work in Australia. So we went about assessing our donors' willingness to essentially provide additional data when they make a blood donation, and also provide blood samples for further research. We published this now a few years ago, and pleasingly, the vast majority, about 90%, 95% or so of our donors said yes. When I go down a little further, though, there was a but, yes, happy to participate in those forms. However, needs to be ethically appropriate, needs to be relevant to what you guys know about. Would you be involved in genetic research or genomic research? Maybe. Yes, if it's related to red blood cell antigens and that sort of side of things, but not necessarily if it's going to have any implications on my genetic propensity for a health outcome. So that's an area that we're sort of working on as well. We've got a number of research projects in that space. So we set the ball rolling. Our aim was really to collect samples and data from around about 6,500 donors across the country to test the feasibility of essentially also putting on top of our donor centres and our processing staff some extra workload, really. We want an extra sample collected. We want a survey carried out and so on. So we did. Just a summary, really said, when we sent out surveys to ask for interest, we had about 25% consented to participate, which is quite a reasonable proportion. Our donors, as I don't need to tell you, are very interested in research, and I think, sitting in a university sending out coal, you might get 3 or 4% response to those sorts of things. So I won't go through all the data, but essentially we're very pleased with the outcome that we got. We looked across the country, sort of staggered the way that we collected these samples across here. These are just the number of samples that we've collected going across from left to right over about a 15-month period, so that we could actually get the system going. Put this into, it shows a lot of extra tubes collected and spun and separated up and stored away. So we've got that system sort of worked through after, for example, getting samples from Perth across to Sydney. I think one break of tubes leaked in the post. So the little things like that, you need to be sort of trying to sort those things out. Then this is really where we're at. So we've got now 6,500 donor samples. We've got apicoat DNA. We're still in the process of extracting DNA from those samples, plasma and serum samples. And then we've sort of got various different systems that we look to make sure that we had a system that we can move on. So really, were our donors representative of the Australian population? Short answer to that was not quite. But once again, this is a feasibility study. I mentioned earlier that about 30% of our population are born overseas. About 23 or 4%, I think it was, of our people who participated in this study were, I think the data's there, 25% in the study. But essentially a lot of this was carried out from a pragmatic point of view as well. So we had to use donor centres where we were able to move into those donor centres. So the next phase of the study really to ensure that we can get a more appropriate demographic distribution across the country is really to target some of those other donor centres. One thing I forgot to mention a minute ago is that just 2 months ago, I think it was, we had a new donor centre in the southern part, southeastern part of Sydney, which has got a very high Chinese, particularly Chinese, but some Southeast Asian as well, community. What we did in opening that new donor centre was take into account that information that I went into the detail about our donors, recruit donors into that particular donor centre. So we've actually got multilingual staff. We've got so it's sort of fitting right into that community. So we're already putting a lot of that work into practice. So our health and lifestyle measures, we do a little bit of data analysis. So I'm looking on there, but in a nutshell, Australian blood donors are slightly heavier than non-donors. Interesting, but don't know why that might be. But they do have a better blood pressure. They don't smoke as much and consume slightly less alcohol. Sort of maybe that's somewhat the healthy donor effect, but I think that's another story and another topic. Blood donors, once again, here we look at the donors took more supplements. Female donors took more iron supplements, which is pleasing to see. The donors had a slightly better dietary and general health measures, but less physical activity, surprisingly. This is comparing our data with a national Australian Bureau of Statistics National Health Survey from a couple of years ago, or a few years ago now. So that's just really where we're at in terms of that blood donor study. Once again, this is something I'm sort of trying to push with our management to become a part of our normal operations, to play a great role in healthcare across the country. So I'll skip that. I mentioned serosurveillance, but I think I might just stop there because I'm chairing and I'll be hitting myself on the knuckle running over time if I don't. So I hope that was of some interest to everyone. As I said, it's sort of broad ranging. I did skip through a lot of stuff that I was planning to put on. Probably pleased I didn't go through that, but more than happy to answer any questions that you might have. Questions?
For your word of mouth study, did you look at, well, I think from my understanding was largely looking at positive word of mouth. Have you thought about sort of negative word of mouth? So the ethnic minority, so sort of.
It's all of the above, essentially. We're really sort of trying to look at what the enablers are and also the sort of restrictors. Well,
I think some it would overweigh just the positives. So anyone who's had a negative experience being referred, for instance, might end up speaking to more people about that negative experience.
Yeah, but I think it's also a case of explaining and educating why those people are referred. That's probably one of the key things that sort of came through as well. It's why.
Yeah.
Sorry.
No, thanks for a really great overview of what you're doing. You mentioned about the feedback of results to and the EDS.
Mm-hmm.
Do you have any plan to feedback results? And
Most definitely, yes. So
what type of results you're planning to feedback?
we won't be anonymised, but essentially the donors all consented to receive information. So they are on a newsletter to provide information on how that's going. So we do have 3 studies that we're actually doing at the moment. One's actually with you, Rebecca, on the familial pseudohyperkalemia project. This is a study of essentially on donors' red cells. So that there's this mutation. Correct me if I get this wrong, but there's a mutation that essentially changes the permeability of red blood cell membranes to potassium, and it's about 4%, I think, of population, something like that. And essentially, red blood cells from those donors will leak potassium once they're stored over time. So it's not a direct impact to the donor, but it's a potential impact to the patient. So we do feedback that information. That's right, I think.
Yeah, we actually, well, in England, and I think you guys are thinking about what your policy is going to be, but so in England, if we identify donors with that variant, we don't necessarily defer them from donation, but we ensure that the red cells are used for adults and not paediatric transfusions, which is the worry that they're getting a really big potassium dose. But so we've been using, in England, we've been using that data to guide our clinical policies, and I think you guys are thinking about the same. So
We certainly tell our donors, and essentially we're trying to convert them to plasma donors.
yeah,
They'll donate, but they can become a plasma donor. That's one of the key things, I guess, the number of donors that we've got. We became a net plasma collector about 4 or 5 years ago now, I think. So it's, the curve of whole blood versus plasma, it's about 20% more of our donors are plasma donors now than whole blood donors.
Forgive me. So I'm just wondering,
Don't have to ask for forgiveness. Ask.
share some data on some, I think it was decreased physical activity in the donors who were in the study, right? So is that compared to the donor population, I'm presuming, in Australia?
Sorry, beginning of it.
Compared to the general population in Australia, they have less physical activity, right? I think you showed.
Yeah.
You know, I'm thinking, is that because like the athletes don't want to give blood? They're like, I don't want to, because the training is. But do you know why that
I
could
think
be?
it's really, the numbers are pretty small. I wouldn't read a lot into
Okay.
it. So we are have been looking potentially at looking at athletes as well. In fact, we do have, it's an interesting point. There's a debate about whether giving blood when you're an athlete is a good thing or a bad thing. Obviously it's a bad thing if you're giving it specifically to sort of blood dope and sort of history, if you like, of the cycle fighters and eco. But I think with we, for example, I don't know if any of you are familiar with Australian rules football. We have in Sydney, Sydney team called Sydney Swans. We actually have and they have red and white jerseys. They're affectionately known, have been known forever as the Bloods because of their red jerseys. They're a great supporter of ours. We have a blood donor centre in the Sydney Swans headquarters at the Sydney Cricket Ground in Sydney. And they're good. They donate as well, but obviously not during season.
Yes, thank you. Large amount of research ongoing. If I can ask a question on first half and not on the donor studies. So I think it was a CHIP study of platelets, indeed a striking result in view of what you had, seen previously.
The CHIPS is the cold stored platelet.
No, the other one. Sorry,
the frozen platelet one. Yeah.
I can't remember the name. It's the frozen platelets one, and so. Non-inferiority is confirmed with a p-value kind of close, okay, but you have secondary outcomes which are, let's say, suggesting, and I think you said that, I'm quoting you, minus 30%.
30%, yeah.
And so, I mean, that's rather substantial. We don't usually see, platelets. That means that platelets are doing something. I'm a bit provocative here, okay? But and because there you're basically comparing two types of platelets, and you're assuming that all you're seeing is due to platelets. So I guess my question is, but that's been checked, obviously. Is there anything else going on for some reason, some imbalance, which could have, in a more potent way, impacted? I guess it's bleeding. I guess some of these secondary.
It's highly probable that there is because they're cardiovascular surgery patients to start off with. They're not in good shape. I think in hindsight, they may not have been the best patients to use in the trial. But I think we're talking about different patients in different studies.
Yeah.
But it's hard to do something like that in a trauma patient, for example.
Yeah. But I guess looking at, so you had a quick, show it very quickly. So there is no obvious imbalance in between groups of randomization, anything,
No, they're almost identical.
because obviously by chance that can still happen, either randomized. Okay. And just associated question, there's a similar study ongoing in New Zealand. Am I
Yes, that's right. Yes.
Okay, so it's going to be interesting to see, when that's coming out?
It is due to complete around about now. I'm not sure. So we completed recruitment around about this time last year. It was around about this time last year. They should be finished. I haven't had an update, to be honest. And separately, there's a study that the US is carrying out, as you know as well, that we think they're going to finish recruiting till the end of this year. But the New Zealand one is essentially the same criteria as ours.
That's fine.
The interesting thing about the New Zealand one is the actual reconstitution of their plasma is different.
Oh,
But I'll go through that with you later.
okay, very good.
That shouldn't have an impact.
Yeah, well, yes, thank you. Well,
Yeah.
once again.
All right. Well, thanks so much, everyone. Oh,
I just have one quick question. As well as you're doing your word of mouth study, it seems like you're not putting all your eggs in one basket here, right? You are doing work on like social, because obviously young people communicate in other ways apart from just speaking to each other, you know, like messaging and things like that. So I'm wondering if there's any work, you're doing any work with that as well.
No, definitely. Like we did some work just some years ago in terms of us messaging and the time of messaging, and when you send every time a donor makes a donation, they get a message to start with, but also when their product is used in a transfusion or shit sent out to a hospital, they get a message that comes back saying your product's just been delivered to this hospital.
Yeah.
So we certainly got that. Yeah, sorry, I glossed over a lot. So we've got about that star diagram. There's about a dozen different projects that cover those various different aspects. I just wanted to go through that one here to give you a little bit of depth.
Okay.
Right, I should move it. I can do it from here, actually, probably. And I'm